We have a stack of heavy reading on the desk today. I'm looking at the 9th edition of Murray's Medical Microbiology, specifically Chapter 14, and we've got Jowett's Chapter 9 open as a cross-reference. And honestly, usually when I pick up a microbiology textbook, I expect a catalog. A list of bugs, a list of drugs, and a list of symptoms.
It's the stamp collecting phase of medical education.
Right. But going through this material on pathogenesis, this doesn't feel like a catalog. This feels like a war room dossier. We aren't just looking at what these bacteria are. We're looking at a strategic playbook of how they invade, how they fight, and how they survive.
That is the perfect way to frame it. I mean, if you approach microbiology as just a list of names, Staphylococcus, Streptococcus, Escherichia, you are going to be miserable. And frankly, you won't be a very good clinician.
Yeah.
Pathogenesis is the story of the interaction. It's a chess match. It's not just about the pathogen's move. It's about the host's defense and the pathogen's countermove.
And the stakes are, quite literally, life and death.
They are. And for the medical student or the learner listening to this is the pivot point. This is where you move from memorizing Staph aureus causes abscesses to understanding why Staph aureus causes abscesses.
Well, why?
We are building a mental model today. We're going to break down the steps of this military campaign. Entry, adhesion, invasion, damage, and evasion.
It's a coordinated sequence. You can't just jump to damage without solving the entry problem first.
Precisely. Virulence isn't a single trait. It's a combination of tools. If a bacterium has a great toxin but no way to stick to your cells, it's harmless.
It's gone.
It just washes away.
So our mission today is to walk through that sequence, step by step.
Yeah.
We're going to deconstruct the bacterial toolkit. We'll look at the grappling hooks, the molecular syringes, the camouflage, and, the heavy artillery.
And we will anchor this in the high-yield material from Murray. If you are prepping for boards, pay attention to the specific mechanisms we discuss. The receptors, the toxins, the secretion systems. That is where the points are.
Okay, let's do it. Let's start at the perimeter. Step 1. Entry. The breach.
The breach. To understand this, you have to visualize the human body not as a welcoming organism, but as a fortress designed specifically to keep things out.
And the outer wall is the skin.
The skin is a formidable barrier. It's not just a wrapper. I mean, think about the environment on the surface of your skin. It is dry.
Right.
It is slightly acidic, and the temperature is lower than the internal body temp. Most bacteria that are adapted to live inside a warm, wet host absolutely hate the skin.
It's a desert to them.
It is a hostile desert. Plus, the outer layer, the stratum corneum, is dead. It's constantly sloughing off. So even if a bacterium lands there, the ground beneath it is crumbling away.
So simply landing on the skin isn't enough to cause an infection?
Generally, no. Murray is very clear on this. The skin is effectively impenetrable to most bacteria unless there is a disruption, a breach in the wall.
Which brings us to the opportunists. We talk about Staphylococcus aureus and Staphylococcus epidermidis a lot.
These are part of your normal flora. They are camping out on the castle walls, surviving that dry, salty environment better than most, but they are waiting for a mistake.
A cut, a scrape, a surgical incision.
Or, in modern medicine, a plastic tube. This is a huge concept for clinical practice. When you insert an IV catheter, a central line, or a prosthetic joint, you are essentially building a highway across the moat.
You're bypassing the wall completely.
You're bridging the sterile interior with the bacteria-laden exterior. Staph epidermidis, which is usually harmless on the surface, loves plastic. It adheres to that catheter and rides it right into the bloodstream.
So that's the skin. But the fortress has gates. We have natural openings, the mouth, the nose, the urogenital tract. These are wet, they are warm, they seem much more inviting.
They are inviting, which is why the defenses there are so aggressive. These are the mucosal surfaces.
Like kill zones.
In a way, yes. You have mucus, which traps bacteria like flypaper. You have IgA antibodies patrolling that mucus. You have lysozyme in tears and saliva that dissolves bacterial cell walls. And then you have the ultimate chemical weapon, the stomach.
The vat of acid.
A pH of 2. That is incredibly corrosive. Most bacteria you swallow are incinerated instantly.
But obviously, food poisoning exists. Salmonella exists. So they have a counter move.
This is the chess match. The enterobacteria, the gut bugs like Salmonella, E. coli, Shigella, are gram-negative. They have a complex outer membrane that acts like armor plating against acid and bile. They can survive the transit through the stomach to reach the intestines.
And there is a numbers game here too, right? The infectious dose.
Yes, absolutely. If you swallow 10 Salmonella bacteria, your stomach acid will probably get them all. If you swallow 1,000,000, a few thousand might survive the acid bath to reach the colon.
Okay.
But compare that to Shigella. Shigella is so acid resistant that you only need to swallow about 10 to 100 organisms to get sick.
That is terrifyingly efficient.
It is. Now, there is a distinction here that Murray makes, and I want to pause on it because it confuses students constantly. The difference between colonization and disease.
Right. Just because the bug is there doesn't mean you are sick.
Exactly. We are colonized by trillions of bacteria. That is our microbiome. Disease only occurs when that relationship creates damage or dysfunction.
The example that stood out to me was Legionella.
Legionella pneumophila. This is a great example of a bacterium that has strict requirements. You can inhale Legionella into your lungs. It has breached the fortress. But if your immune system is competent, or if the temperature isn't right, it doesn't replicate effectively.
So it's there, but it's not thriving.
It enters, but it doesn't conquer. No disease.
Contrast that with the cystic fibrosis patient.
This is the clinical correlation you need to know. In a healthy lung, you have the mucociliary escalator. Millions of tiny hairs, the cilia beating in unison to sweep mucus up and out of the airways. It keeps the lungs sterile.
The conveyor belt to the throat.
Right. In cystic fibrosis, the genetic defect makes the mucus thick, dehydrated, and sticky. The conveyor belt jams, the mucus just sits there.
And bacteria love stagnant pools.
It becomes a petri dish. Staphylococcus aureus or Pseudomonas aeruginosa enter, and instead of being swept out, they find a nutrient-rich, protected environment. They colonize. And because they can grow unchecked, they cause massive inflammation and tissue destruction. That is disease.
So the lesson is, entry is just the first hurdle. But once you are in, you are facing a new problem. The body is constantly trying to wash you away. Urine flow, gut peristalsis, coughing. It's like trying to swim up a raging river.
Which brings us to step 2. You have to anchor yourself. Adhesion.
The Velcro phase.
If you don't stick, you don't stay. Evolution has driven bacteria to develop highly specific molecular tools to grab onto host tissues. We call these adhesins.
And the most famous of these, the one that comes up on every exam, is the fimbriae, or pili.
Think of these as long, hair-like grappling hooks extending from the surface of the bacteria. E. coli is the poster child for this.
But not just any E. coli. We need to talk about the ones that cause kidney infections.
Right. Uropathogenic E. coli. Now listen to the engineering here. The bladder is a high-flow environment. When you void, you are flushing the system with significant force.
Right.
To stay there and to climb up the ureters to the kidneys, E. coli uses P. fimbriae.
P. fimbriae. Why P?
It stands for pyelonephritis, or the P blood group antigen. At the very tip of this long hair-like structure, there is a receptor-binding protein. It is shaped perfectly to fit into a specific sugar molecule on the surface of your uroepithelial cells.
The Galgal receptor.
Galactus galactus. This is a specific molecular handshake. The P. fimbriae locks onto the Galgal receptor on the cells lining the upper urinary tract.
And this is why some people are more prone to UTIs than others?
Partially, yes, but it explains the clinical presentation. A generic E. coli without these pili might cause a little bladder irritation, cystitis, or be washed away. But an E. coli with P. fimbriae has the equipment to climb upstream, lock onto the kidney tissue, and cause pyelonephritis, a serious high-fever kidney infection.
So the structure dictates the disease.
Structure dictates function. Structure dictates disease. It's a perfect example.
Now, gram-negatives use these hairy pilis. But gram-positives, like staph and strep, they have a different approach. Murray talks about MS-grams.
A terrible acronym. Microbial surface components recognizing adhesive matrix molecules.
It rolls right off the tongue.
It's basically surface glue. Instead of looking for a receptor on a cell, these bacteria look for the stuff between the cells, the extracellular matrix.
The connective tissue.
Right. Fibronectin, collagen, laminin. Staphylococcus aureus is covered in proteins that bind to fibronectin and collagen. This is why it is so good at infecting wounds. If you have a cut, the connective tissue is exposed. Staph sees that exposed collagen and latches on immediately.
So E. coli grabs the cell, staph grabs the foundation.
That's a good way to visualize it, yes.
There is a third adhesion mechanism that we need to spend some real time on because clinically it is a nightmare, the biofilm.
The biofilm. This is less like Velcro and more like building a bunker.
Or a slime city.
A slime city is accurate. Here's how it works. Free-floating bacteria, we call them planktonic, are vulnerable. Antibiotics can hit them, immune cells can eat them.
Hmm.
But some bacteria, particularly Pseudomonas aeruginosa and Staph epidermidis, have a social intelligence.
They can talk to each other?
Chemically, yes. It's called quorum sensing. As they attach to a surface, say a hip replacement or a catheter, they secrete small signal molecules. As the population grows, the concentration of these signals rises.
They're counting their numbers.
Exactly. When the signal hits a specific threshold, it triggers a massive genetic shift in the whole colony. They stop swimming, they shut down their flagella, and they start pumping out a thick, sticky polysaccharide goo.
The matrix.
The exopolysaccharide matrix. It encases the bacteria in a protective dome.
And once that dome is up, good luck treating it.
It is incredibly difficult. This is a huge issue in orthopedics. If you have a patient with a prosthetic hip joint that gets infected with a biofilm-forming Staph epidermidis, you can dump antibiotics into that patient's blood. But the drugs physically cannot penetrate the slime layer to reach the bacteria in the center.
It's a force field.
And the bacteria deep inside the biofilm go dormant. They slow down their metabolism. Since most antibiotics work by attacking actively dividing cells, these dormant persister cells are immune.
So what's the solution?
Often, you have to rip it out. You have to surgically remove the infected hardware, wash out the wound, and start over. You can't kill the bunker, you have to remove the ground it stands on.
That is brutal, but effective.
It's the only way sometimes.
Okay, so we've breached the wall and we've anchored ourselves. Now, for some pathogens, staying on the surface isn't enough. They want to get inside the VIP room. They want to invade the host cells. Step 3.
Invasion.
This is where the biology gets really sci-fi.
Yeah.
I was reading about the type 3 secretion system in Jawets, and it is honestly mind-blowing. They call it a molecular syringe, but it's more complex than just a needle.
It is a nanomachine. The type 3 secretion system is found in many gram-negative pathogens. Salmonella, Shigella, Yersinia, E. coli. The bacteria builds a needle-like structure that extends from its surface, punches a hole in the host cell membrane, and injects bacterial proteins directly into the host's cytoplasm.
It's injecting its own software code into our hardware.
Yes. We call these effector proteins, and their job is to hijack the host cell's controls.
Specifically the skeleton, right? The cell skeleton.
The actin cytoskeleton. Normally, the cell maintains a nice round shape, but these bacterial proteins force the actin to polymerize rapidly and chaotically at the site of injection.
So the cell membrane starts freaking out.
It ruffles, it throws out these waves of membrane that wrap around the bacterium. It's like the bacteria is commanding the cell, hug me, pull me in.
Forced phagocytosis.
Exactly. The host cell, which isn't a phagocyte, it could be an epithelial cell lining your gut, is forced to engulf the bacteria.
Why do they want to be inside?
Inside is safe. Antibodies can't reach you inside a cell. White blood cells can't find you. And the cytoplasm is full of nutrients. It's a safe house.
And Salmonella uses this to cross the gut lining, right?
Yes. Salmonella targets specific cells in the colon called M cells.
Microfold cells.
M cells are the surveillance outposts of the gut immune system. They sit over Peyer's patches. Their job is to sample gut bacteria and hand them to immune cells. Here, check this out.
Salmonella hacks the outpost.
Salmonella uses its type 3 syringe to force the M cell to take it up. It rides the M cell like an elevator through the mucosal barrier and exits into the tissue underneath. It uses the body's own surveillance system as a backdoor entrance.
That is devious.
It is evolutionary genius.
Okay, we are in. We are safe.
Huh.
Now comes the messy part, the damage. Step 4. This is where we talk about the weapons. Toxins.
And for the student, this is the highest yield section of the entire chapter. You will see questions on toxins for the rest of your career. We need to divide them into two massive categories: exotoxins and endotoxins.
I like the analogy. Snipers versus shotguns.
It holds up. Exotoxins are the snipers. They are proteins secreted by the bacteria, gram-positive or gram-negative, that travel to a specific target and execute a precise biochemical hit.
Endotoxin.
Endotoxin is the shotgun. It is a structural part of the bacteria itself, and when it's released, it causes a massive, systemic, indiscriminate explosion of inflammation.
Let's look at the snipers first. The exotoxins. And the classic design here is the AB toxin.
The binary weapon, it has two components. The B subunit is for binding. It's the delivery vehicle. It finds a specific receptor on the target cell.
Right.
The A subunit is for action. It's the toxic enzyme.
So B gets you through the door, A does the dirty work. Let's run through the Hall of Fame here. Diphtheria toxin.
Corynebacterium diphtheriae. The B subunit binds to heart and nerve cells. The A subunit enters, and its target is elongation factor 2, or EF2.
Elongation factor implies it's making something longer.
It's essential for protein synthesis. The ribosome needs EF2 to build proteins. Diphtheria toxin freezes EF2.
So the protein factory shuts down.
Completely. And a cell that cannot make proteins dies. That is why diphtheria causes that necrotic pseudomembrane in the throat. It's a layer of dead host cells. And if it hits the heart muscle cells stop maintaining themselves and die. Myocarditis.
Deadly efficiency. Okay, next one. Cholera.
Vibrio cholerae, totally different mechanism. The target is the gut lining. The A subunit enters and modifies a G protein.
G proteins are the switches for cell signaling.
Right. Cholera toxin locks the stimulatory G protein in the on-in position. This constantly activates adenylate cyclase,
Which cranks out CAMP.
massive amounts of cyclic AMP. Now, high CAMP tells the cell, dump chloride. The cell pumps chloride ions out into the gut lumen.
And basic physics osmosis tells us that water follows salt.
So water floods out of the cells and into the intestines to chase that chloride. You get liters and liters of watery diarrhea, the rice water stool. Notice the cell isn't dead. The factory is still running, but the control panel is hot-wired to flush.
So diphtheria kills the cell, cholera just hacks the plumbing.
Correct. That's a great way to put it.
Now the neurotoxins, this is my favorite comparison because it's so elegant. Tetanus versus botulism.
The yin and yang of paralysis. Both are Clostridia species. Both are anaerobes. Both make AB toxins that are proteases. And they both target the same machinery, the snare proteins.
Snare proteins are the docking clamps that allow neurons to release neurotransmitters.
Right. No snares, no signal release, but the location is the difference.
Let's walk through tetanus. You step on a rusty nail, Clostridium tetanus spores get in.
The bacteria stay in the wound, but they release tetanospasmin. This toxin travels retrograde backwards up the motor neurons all the way to the spinal cord.
It goes to the command center.
And specifically, it targets the inhibitory interneurons, the Renshaw cells. These are the cells that tell your muscles to relax. They release glycine and GABA.
So tetanus toxin cuts the snares in the relaxed neurons.
Exactly. You lose the brakes, your motor neurons are firing, contract, contract, and there is no one to say stop.
So you get rigid, spastic paralysis, lockjaw, the arched back.
It's agonizing. You are flexing every muscle in your body until the bones break.
Now flip it. Botulism. Clostridium botulinum.
Botulism toxin works peripherally. It stays at the neuromuscular junction where the nerve meets the muscle. It cuts the snares in the motor neuron itself.
The neuron that releases acetylcholine.
And acetylcholine is the go signal. It's the contract signal.
So tetanus cuts the brakes, botulism cuts the gas line.
Perfect analogy. In botulism, no acetylcholine is released. The muscle never gets the message to move. You get flaccid paralysis, floppy baby syndrome. You stop breathing because your diaphragm literally cannot flex.
Two toxins, same molecular mechanism, just cutting snares, but opposite clinical effects purely based on where they strike.
It is biological sniper fire. It's beautiful in a terrifying way.
Okay, those are the snipers. But there's another class of exotoxins that are, well, they're the riot starters.
Yeah.
The superantigens.
This is a concept that students often struggle with, but it's crucial for understanding toxic shock syndrome.
Normally, T-cell activation is very specific, right?
Extremely. It's a lock and key. An antigen-presenting cell shows a tiny piece of a germ on its MHC tie or receptor. A T cell with the matching receptor sees it. They shake hands. Okay, we found the bad guy. This activates maybe 1 in 10,000 T cells.
Precision strike.
A super antigen like TSST1 from Staphylococcus aureus breaks the rules. It doesn't fit in the lock. Instead, it grabs the MHC-TI molecule on one side and the T cell receptor on the other, and it handcuffs them together.
It forces the handshake.
It forces the interaction, regardless of whether they match. And it binds to the beta chain of the T cell receptor. Because many T cells share this beta chain structure, the superantigen activates up to 20% of all T cells in the body simultaneously.
From 1 in 10,000 to 1 in 5?
Yeah.
That is a massive escalation.
It's a nuclear event. All those T cells start screaming at once. They release cytokines IL-1, IL-2, TNF-alpha.
The cytokine storm.
Exactly. And that storm causes the symptoms. High fever, low blood pressure, or shock, rash, multi-organ failure. The bacteria isn't eating your liver. Your immune system is frying it in a panic response.
Okay, one last group of exotoxins before we switch to the shotgun. The membrane disruptors. These seem less subtle.
These are the heavy artillery. Clostridium perfringens produces alpha toxin. This is a lecithinase.
It sounds like an enzyme that eats lecithin.
And lecithin is a major component of cell membranes. This toxin literally dissolves the cell walls of your tissue. It turns muscle into mush.
Gas gangrene.
Yes. It creates that crepitus or gas pockets in the tissue. It's liquefactive necrosis. No finesse, just destruction.
Okay. We have covered the proteins. Now, the main event for gram-negatives, the shotgun, endotoxin.
Endotoxin is fundamentally different. Exotoxins are secreted. Endotoxin is the bacteria. Specifically, it is lipopolysaccharide, or LPS.
This is a gram-negative exclusive.
Correct. Gram-positives have a thick wall, but no outer membrane. Gram-negatives have that outer armor we talked about earlier. LPS is the main structural brick of that armor.
So when does it become a weapon?
When the bacteria dies, or when it divides rapidly. The cell wall breaks up, and LPS is released into your bloodstream.
And the toxic part is lipid A.
Lipid A. This molecule is the alarm signal that our immune system has evolved to fear. We have a specific receptor on our macrophages called TLR4.
Toll-like receptor 4?
TLR4 is designed to spot lipid A. When it binds, the macrophage doesn't just get active, it goes nuclear. It releases the acute phase cytokines, IL-1, IL-6, and TNF-alpha.
We keep hearing these names. IL-1 gives you fever, TNF-alpha. That's the scary one.
Tumor necrosis factor alpha. It causes your blood vessels to leak. Fluid moves from your blood into your tissues. Your blood pressure crashes. That is septic shock.
And LPS also messes with clotting.
Yes. It activates the coagulation cascade. You start forming tiny clots in all your small blood vessels. This uses up all your clotting factors.
So you clot in the capillaries.
And you bleed out everywhere else. Disseminated intravascular coagulation, DIC, it is a catastrophic failure of the body's homeostasis.
And this is why treating gram-negative sepsis is so tricky. If you give a massive dose of antibiotics and kill all the bacteria at once,
you might release a massive wave of LPS all at once and actually worsen the shock initially. It's the Jarisch-Herxheimer reaction principle, though that's technically for spirochetes the concept holds. You are releasing the toxin by killing the bug.
that is the shotgun.
Yeah.
Messy, systemic, dangerous.
Exactly.
We have covered a lot of offense, but the bacteria has to survive long enough to do all this. It needs to defend itself. Step 5, evasion, the camouflage.
The immune system is a hunting machine. Neutrophils and macrophages are constantly patrolling, looking to eat anything that doesn't belong. The bacteria need to hide. The first and most important tool is the capsule.
The slippery germ.
Imagine you are trying to tackle a football player, but he is covered in a thick layer of grease. That is a capsule. It's a gelatinous polysaccharide layer that covers the bacteria. Neutrophils try to grab it with their receptors, but they just slide off.
It prevents phagocytosis.
Effectively, yes. It also covers up the bacterial surface markers, the PMPs, so the immune system doesn't even recognize it as a threat immediately.
There is a vital clinical connection here with the spleen.
This is high yield. The spleen is the organ where the blood is filtered, and it is the primary site for clearing encapsulated organisms. The spleen has a specialized way of trapping and opsonizing or tagging these slippery bugs.
So if you don't have a spleen,
If you are a splenic, maybe from trauma or sickle cell disease, you are incredibly vulnerable to sepsis from encapsulated bacteria.
the Sheehan organisms.
Streptococcus pneumonia, Haemophilus influenza, Neisseria meningitis. If you have a patient without a spleen, they need vaccines against these three specifically because they have lost their main defense against that capsule.
Some capsules are even sneakier. Look at Streptococcus pyogenes group A strep,
It makes a capsule out of hyaluronic acid.
which sounds familiar because it's in my face cream.
It's in your connective tissue. It is a normal human substance. By covering itself in hyaluronic acid, the bacteria is effectively wearing a human skin suit. The immune system bumps into it, says, oh, just some connective tissue here, and moves on.
Molecular camouflage.
It's brilliant.
What if you can't hide on the outside? What if you get eaten?
Then you hide on the inside. Intracellular survival. We touched on this with invasion, but some bugs make a lifestyle out of it. Mycobacterium tuberculosis is the master.
TB gets inhaled into the lung.
Mm.
The alveolar macrophage eats it. That should be game over.
Normally, the macrophage takes the bacteria into a bubble called a phagosome. Then it fuses that bubble with a lysosome, a bag of acid and enzymes. Phagolysosome fusion. It digests the bug.
But TB stops the merger.
TB secretes proteins that inhibit the fusion. It sits inside a phagosome, comfortable, protected from antibodies, protected from acid, and just replicates. It turns the prison cell into a luxury condo.
And then there is Listeria monocytogenes.
Yeah.
It takes it a step further.
Listeria is the escape artist. It gets eaten, it's in the phagosome, but it secretes a pore-forming toxin called Listeria licino.
It bursts the bubble.
It dissolves the phagosome membrane and escapes into the cytoplasm. Now it is swimming in the nutrient-rich center of the cell, but it doesn't stop there.
The rocket tail.
This is one of the most cinematic images in microbiology. Listeria recruits the host cell's own actin molecules. It polymerizes them behind it to create a propulsive tail.
Like a motorboat engine.
It zooms around the cytoplasm, picks up speed, and slams into the cell membrane. It pushes so hard it creates a finger-like projection that pokes into the neighboring cell. The neighbor engulfs it, and boom, Listeria is in the next cell without ever entering the extracellular space.
It tunnels from room to room without ever going in the hallway.
Exactly. It never exposes itself to antibodies in the blood.
Wow. Okay, one more evasion tactic. Changing your face. Antigenic variation.
Neisseria gonorrhea is the champion here. We talked about pili earlier. Pili are proteins. They are antigens. The immune system sees them and makes antibodies to attack them.
So you develop immunity.
You should. But the gonococcus has a trick. It has one active gene for the pilus and a library of silent gene cassettes with different sequences.
It is a wardrobe of disguises.
It constantly shuffles these genes through recombination. By the time your body makes IgG antibodies against pilus type A, the bacteria has already switched and is expressing pilus type B. The antibody is useless.
It's a shapeshifter.
It explains why there is no vaccine for gonorrhea and why you can get it over and over again. The target is constantly moving.
Finally, step 6. Sometimes the bacteria doesn't kill you. You kill you. Immunopathogenesis.
Friendly fire. This is a crucial concept. We tend to think of symptoms as being caused by the bug's toxins, but often the tissue damage is a result of our own immune response trying to clear the bug.
TB is the classic example here too.
Granulomas. The immune system can't kill the TB bacteria hiding in the macrophages. It gets frustrated, so it calls in reinforcements. T cells, macrophages, giant cells, they surround the infected area to wall it off.
Containment protocol.
That is a granuloma. It traps the bacteria. But that granuloma takes up space, it replaces healthy lung tissue, and if the cell's in the center, dicaceous necrosis, it creates a cavity. The lung is being destroyed not by the TB toxin, but by the immune system's siege warfare.
And then there is post-infection autoimmunity, rheumatic fever.
This is a tragedy of molecular mimicry. You get a throat infection with group A strep, S. pyogenes, you recover, but the bacteria has a protein on its surface called M protein.
And M protein looks familiar.
M protein has a molecular structure that looks suspiciously like myosin, a protein in your heart valves.
So the antibodies you made to kill the strep.
They start cross-reacting. Weeks after the throat infection is gone, those antibodies are circulating, and they start attacking your own heart valves. Rheumatic heart disease.
Strep licks the joints but bites the heart.
Precisely. The bacteria is long gone. The damage is done by your own immune memory.
This is dense. We have covered entry, adhesion, invasion, damage, evasion, and friendly fire. It is a complete military doctrine.
It is. But if you hold this framework in your head, the individual bugs stop being random facts. They become logical players in this game.
Okay, let's verify that logic. It is time for the rapid fire quiz. I'm going to throw a few at you. You ready?
Let's do it.
Question 1. A young woman presents with symptoms of pyelonephritis, high fever, flank pain. Which specific bacterial structure is most likely responsible for the organism's ability to ascend to the kidney?
Okay, so that's pyelonephritis. We're thinking urinary tract. The answer has to be the P. fimbriae, or P. pili of E. coli. Remember, generic pili might cause cystitis, but the P. fimbriae bind the gal-gal receptor in the kidney, allowing the bacteria to fight the flow of urine and ascend.
Got it. Question 2. We have a patient with spastic paralysis, lockjaw, and muscle rigidity. The toxin responsible functions by cleaving snare proteins in which specific cell type?
Spastic paralysis is the key. That points straight to tetanus. The toxin travels retrograde to the spinal cord and hits the inhibitory interneurons, specifically the Renshaw cells. By blocking release of glycine and GABA, the relax signals, you get that rigidity.
Perfect. Question 3. A patient in the ICU developed septic shock from a gram-negative rod. What is the specific molecular component of the bacteria that triggered the TLR4 receptor on the macrophages?
Gram-negative septic shock means we're talking about LPS. And the specific part of LPS that our TLR4 receptor recognizes is lipid A. That's the toxic anchor that kicks off the whole cytokine storm.
Excellent. Question 4. Staphylococcus aureus is coated in protein A. How does this surface protein help it evade the immune system?
Protein A is a classic immune evasion tactic. It binds the Fc region of IgG antibodies. It holds the antibody backwards so the handle that phagocytes would normally grab is covered up. It prevents opsonization.
Awesome. Question 5. A pregnant woman eats unpasteurized cheese and develops a listeria infection. Listeria moves from cell to cell without entering the extracellular space. What host protein does it manipulate to do this?
Oh, the rocket tails. That's Listeria's signature move. It hijacks the host's actin. It polymerizes actin filaments behind it to propel itself through the cytoplasm and punch into the next cell.
Love it. Okay, last one. Question 6. Superantigens like TSST1 cause massive immune activation. They bind to the MHC2I molecule and what part of the T cell receptor?
This is the riot starter mechanism. They bypass the normal antigen recognition by binding to the beta chain of the T cell receptor, or TCR, on the outside of the antigen binding groove. That's how it cross-links everything and activates a huge percentage of T cells.
How did you do? If you stumbled on the toxins, go back to the tables in Murray chapter 14. That is definitely high-yield territory.
And if the secretion systems were fuzzy, Jowett chapter 9 has some great diagrams on the type 3 syringe.
So looking at this whole picture, what is the big takeaway?
The takeaway is that pathogenicity is not accidental. It's complex, it's coordinated, and it's expensive for the bacteria. They don't just happen to cause disease. They have evolved specific tools to overcome specific barriers.
And recognizing those tools gives us targets.
Exactly. If we know the pull structure, we can make a vaccine to block it. If we know the toxin mechanism, we can make an antitoxin. Understanding the mechanism is the first step to winning the war.
And remember, despite all these weapons, the bacteria isn't trying to be evil.
No, not at all. A dead host is a dead end. From an evolutionary perspective, killing you is a failure. They just want to survive and replicate. The disease, the fever, the shock, the damage, that is often just collateral damage in their fight for existence.
Collateral damage. It changes how you see the infection. Thanks for diving deep with us. Keep asking why. Keep building those mental models, and we'll see you on the next deep dive.
See you then.